The single chapter Enzymes accounts for some of the most commonly lost marks in Cambridge A level biology. Not because it's hard, but because of one missing key term. Let's get started with what are enzymes or describe enzymes. So, enzymes, as we all know, are biological catalysts. The word biological is coming from enzymes are globular protein. They have complex 3D tertiary structures, which give them those specific shapes, making an enzyme specific to a substrate. Catalysts, just like chemistry catalysts, they speed up the reaction without being used up. The fun fact is that almost all the metabolic reactions, they are catalyzed by an enzyme. Based on their function, enzymes can be of two types, intracellular and extracellular. So intracellular enzymes, they are secreted and made within the cell and they act within the cell, just like DNA polymerase for DNA replication. However, extracellular enzymes are the ones which are secreted outside the cell and they act in the cavities of the body, like all digestive enzymes. Amylase, protease, lipase, all of them, they are all extracellular enzymes. Moving on to how do enzymes function, we all know that every chemical reaction requires energy to convert reactants to products, and it is usually very high. One way of reducing the activation energy is by increasing the temperature, which we do in our chemistry experiments. But in body, that's not viable. Enzyme lowers the activation energy significantly, and thus they speed up the reaction. But the question is, how do enzymes lower the activation energy? Well, when the enzyme binds to the substrate to weaken its intramolecular bonds, thus facilitating the conversion of reactant to products. Coming to the structure of an enzyme, so each enzyme has got this active site which is complementary to the shape of substrate. Now the key for you to remember is the shape is complementary. The shape is not identical, the shape is not similar, not same, it is complementary. So that's a key word for you to remember. This enzyme substrate complex which is formed is a temporary structure. It's not permanent. So once the reaction is complete, enzyme is going to release the products and enzyme would be ready to be reused up in another reaction. Now coming to the working models of enzymes, we all know the conventional lock and key model. Well, it is quite simple, very straightforward, and it explains how the specificity of the enzyme to a specific substrate works. It makes all the sense. But the problem here is that enzymes aren't rigid. Remember, they are globular proteins. They have three-dimensional structures. They have hydrogen bonds, which are flexible. So enzymes clearly are not rigid. An active site, for that reason, cannot be rigid and fixed. This gives rise to what we call as induced fit model, which is the modern hypothesis, where the active site is flexible and not rigid. As the enzyme approaches the substrate, the enzyme is going to mold itself around the substrate. Just like when you have a glove and you put your hand in, the glove is going to stretch a bit to take the shape of your fingers to give you a perfect fit. Just like that, enzyme molds around the substrate to give a perfect fit for it. This is the currently accepted model because it very well explains how enzyme inhibitors work. Now, what kind of reactions do enzymes act upon? Well, they act on any kind of reaction. They can be breaking down a substrate into products, or they may be joining a couple of substrates to form one product. So, synthesis or breakdown, anything is possible. Usually, we get to know what the enzyme is doing by its name, like reductase will cause reduction, oxidase will cause oxidation, polymerase is like a synthesis kind of an enzyme. We are next moving to the factors affecting enzymes. And the first factor that we'll be discussing is temperature. We know from the kinetic molecular theory, as the temperature is increasing, the kinetic energy of molecules increases, and there are more collisions within the molecules. Okay, which leads to increased rate of reaction. Similarly, when an enzyme is exposed to higher temperature, as we are increasing the temperature slowly, the molecules gain kinetic energy and they collide into each other more often. And the orientation of the enzyme and the substrate is better aligned to each other. Thus, more enzyme substrate complexes are formed, and hence the rate of reaction increases. This continues till a certain temperature, at which the rate is maximum. This temperature we call as optimum temperature. Why is the rate maximum here is because the enzymes are in just accurate orientation to be able to bind to the substrate. So the maximum enzyme substrate binding happens at this stage. As the temperature is increased further, the hydrogen bonds and the non-covalent bonds, they start ripping apart, and this distorts the tertiary structure of this globular protein. The enzyme get distorted, the active site is deformed, and the enzyme can no longer bind to the substrate. This deformation of the enzyme structure is called denaturation. And enzyme denaturation is unfortunately permanent. We cannot reverse it. So once the enzyme is denatured, it's done. We cannot reuse it. We cannot replenish it back. A similar effect is there of pH on the denaturation of the enzyme. Optimum pH of an enzyme is determined by the amino acid present in the tertiary structure of the enzyme. Some enzymes act at an acidic pH and some enzymes act at an alkaline pH. For example, we know pepsin is secreted in our stomach and its optimum pH value is 2. However, amylase, which is secreted in the saliva in our mouth, the optimum pH value is 7 for it. If you deviate from the optimum value, the same thing happens. The hydrogen bonds get broken because the hydrogen ions of the pH value we are in, they are going to interfere with the hydrogen bonds and the ionic bonds, and this is going to distort the tertiary structure of the enzyme. The active site will change shape and the reaction rate will fall. Okay, the last factor that we'll be considering is the effect of substrate concentration. Now, obviously, if there is more substrate, be more enzyme and substrate, complexes form and the reaction will keep increasing. And this will continue till a certain substrate concentrations where the substrate has occupied all the active sites of the enzymes. So because the enzyme concentration is limited in our body, adding more substrate will increase the rate of reaction to a certain limit. When all the active sites of the enzymes are occupied, and then we reach the plateau phase. At this, the rate that we have reached is the maximum rate, and we call it the Vmax, as all active sites are occupied, and this is our maximum possible rate of reaction. The Michaelis Menten constant can be figured out for any enzyme using the Vmax of it, and this constant relates to the affinity of the enzyme for a substrate. So lower the value of Km, the Michaelis Menten constant, higher the affinity of the enzyme for the substrate. So these calculations are very helpful in development of medicines or inhibitors, whatever we need to target the action in the body. Finally, we come to enzyme inhibitors, which I've been mentioning again and again since the induced fit model. As the name suggests, the enzyme is getting inhibited. So we don't want the enzyme to function. We have two categories into it, competitive inhibitor and non-competitive inhibitor. So if you can see in the picture, the blue one is the enzyme and the red one is the substrate. So the shape of the substrate and the enzyme is complementary to each other. The yellow one is inhibitor. Now inhibitor is also complementary to the enzyme. So inhibitor and the substrate, they compete with each other to get bonded to the active site. If we have a lot of inhibitor concentration, of course, the inhibitor will outcompete the substrate and not let it bind. It's going to occupy all the active sites of the enzyme. So substrate has no place left to bind. A common example for it is penicillin antibiotic, which acts on the bacterial enzymes, which help in the cell wall production. So penicillin, when we take, it is basically acting on bacterial enzyme, not allowing the enzymes to form cell wall. And without cell wall, bacteria cannot survive. So bacteria growth and reproduction is affected, and this is how we get rid of the infection. The other category is non-competitive inhibitors. And if you look at the picture, the yellow inhibitor in this case is nowhere close to the active site. Rather, it is binding to other part of the enzyme, which we call as an allosteric site, which is not active site, the other side of the enzyme. When the inhibitor binds to the allosteric site, it kind of changes, it strains the bonds within the enzyme structure, and it changes the overall shape of the enzyme, thereby changing the shape of the active site also. Now, the substrate is no longer complementary to the active site, and that's why the enzyme substrate complex cannot be formed. In this way, the non-competitive inhibitor also inhibits enzyme function. A common example is cyanide poisoning. So cyanide is a chemical which affects the cytochrome C oxidase in our body, which is an essential respiratory enzyme. Now, the question comes, can increasing substrate concentration reverse the inhibition? Well, for competitive, yes. You add more substrate, the substrate will outcompete the inhibitor and start binding to active site more. So, yes, the competitive inhibitors can be reversed by increasing the substrate concentration. But if we look at the non-competitive inhibitor, even if we keep adding more substrate, the active site shape is changed, so the inhibition cannot be reversed by increasing substrate. However, that doesn't mean that non-competitive inhibitor is like a permanent change in the enzyme. This is going to last only till the time inhibitor is in the body. Eventually, all these molecules are going to be eliminated, excreted out of the body, and then the enzymes will be ready to function again like normal. So, both the inhibitions are reversible. They work only till the inhibitor is present in the body. But if we ask about can substrate concentration increase, reverse the inhibition? Well, it's a yes for competitive and no for non competitive. Lastly, we reach cofactors and coenzymes. As the name suggests, there is co in it. So they work in coordination, they are cordially related to the enzyme. These are certain molecules which are necessary for the enzyme to function. We have two categories for them. Cofactors, they are inorganic molecules, so they are not made of carbon but other elements like zinc, magnesium, iron, and they help shape the active site and assist in the reaction. While coenzymes are organic molecules, so they are carbon based molecules, but they are not proteins. So, usually, all the vitamins, vitamin B, vitamin C, they are coenzymes. And some other examples are NAD and NADH, which is a common conversion during the respiratory cycle. And with this, we wind up our topic. You have got enzymes, and now it's time for you to start practicing past papers because that's where you are able to self assess yourself. If you have any question that you need help in, feel free to put a comment in this video and I'll respond back. Thank you.